Welcome everyone. Today we are going to learn about mobility of lipids in the plasma membrane. In 1970, fluid mosaic model was proposed. The term fluid is because of the mobility of the lipid molecules in the plasma membrane. Now, also the proteins are mobile, but in this video we are focusing on mobility of lipids. So today's topic is mobility of lipids in the plasma membrane. Now there are four different movements are performed by the lipid molecules. The first is rotation, second is flexion, third is lateral diffusion, and fourth is flip-flop movement. Okay, here these three movements like rotation, flexion and lateral diffusion occurs in a particular leaflet. Now what is leaflet of plasma membrane? In my previous video I have mentioned that plasma membrane is actually bilayer in structure. So there are two lipid bilayer present, two lipid layers present. The outer layer, this layer is called exoplasmic leaflet, and the inner layer is called cytosolic leaflet. Now, as these inner leaflets face the cytosol of the cell, that's why it is called cytosolic leaflet, and the outside is called exo. Exo means outer side, exoplasmic leaflet. Now, these three movements, rotations, flexions and lateral diffusions occurs in a particular leaflet. That means it can occur in the exoplasmic or cytosolic leaflet, in both leaflets. But flip-flop movement means here the lipid molecules will migrate from exoplasmic to the cytosolic, or some lipids can migrate from cytosolic to the exoplasmic leaflet. So this is the migrations of lipid molecules. So these three movement occurs in the particular leaflet of the membrane, but flip-flop occurs. Flip-flop movements means migration of lipid molecules from one leaflet to the other leaflet. Now let's talk about each and every movement in more details. Rotation means here, let's suppose this is the head group of the lipid molecules present in the plasma membrane, and this is the two fatty acid chains attached with the head group. Rotation means here thermal motion actually helps these lipid molecules to rotate around their own axis. So it can rotate around their own axis. One biophysical techniques can measure the rotation of the lipid molecules called electron spin resonance, or in short we call ESR spectroscopy. So by these biophysical techniques you can measure the rotation of the lipid molecules. I'm not going into details of this technique. Now flexion means the lipid molecules, the fatty acids that is attached with the polar head group is actually flexible, so it can move away and comes close. Let's see. Suppose this is the position of the two fatty acids. Now the distance between two fatty acids is like this, but these fatty acids can actually move away from each other. So the distance between the two fatty acids can increase because they are flexible, so they can move or come close. And this movement is called flexion. This movement is called flexion. So rotation, flexions can occur in a particular leaflet. Also lateral diffusion. Now lateral diffusion means, let's suppose this is the one leaflet of the plasma membrane, and here one lipid molecule is present. Let's suppose this is the lipid molecule present in a particular leaflet. It could be exoplasmic or cytosolic. Now this lipid molecule can move in any directions in the plasma membrane or the leaflet, in any directions. Okay, this is called lateral diffusion. It has been found that in case of natural plasma membrane or artificial lipid bilayer that I mentioned in my previous video, liposomes, in both these situations, these lipid molecules can exchange with its neighbor lipid molecules the 10 to the power 7 times per second. See how fast it can move these lipid molecules from its lever molecules. It can change the place. Okay, now let's talk about this lateral diffusion in more details. One technique can measure the lateral diffusion. It can quantify how much lateral diffusion is occurring in the plasma membrane. The technique is called, very important technique, remember the name, fluorescence recovery after photobleaching. Now the technique actually tells the whole story. Here, fluorescence recovery after photobleaching, in short we call FRAP, FRAP. This technique actually is performed in 3 steps. The first step is cells are stained with fluorescently tagged antibody. So this is the first step. What will happen? Suppose, let's remove this plasma membrane, these lipid molecules. Suppose this is the leaflet of a plasma membrane, and here lipid molecules are actually present. I'm just showing one row of lipid molecules. Let's suppose. Okay, so all over the leaflet lipid molecules present. I'm just showing few lipid molecules to actually show how these actual techniques occur. So what we'll do at first, we will stain the cell with fluorescently tagged antibody. What is fluorescent tagged antibody? Let me explain. Let's suppose this is an antibody. Okay, if this is an antibody, this is the Fc portion of the region, Fc. This is the whole Fc portion of the antibody. Now, a fluorescent tag is actually attached with the Fc portions of the antibody here. It could be red, green, or any color. So let's suppose this is green in color, so the fluorescent tag will be FITC. So if we use FITC tag, it will fluorescent red or green signals. So this is called fluorescent tagged antibody. Now if we stain the cells with the fluorescent antibody, it will bind with the lipid molecules. Obviously this antibody should bind with the particular or lipid molecules. If we target this antibody with the lipid molecules, it could be phosphoglycerides or phospholipids or sphingolipids, then we can bind, this antibody can bind and it will fluoresce. Let's see in another way. Let's suppose this is the cell and we have stained these cells with fluorescent tag antibody. Then we will find fluorescence coming from all over the cell membrane. Okay, all over the cell membrane it will fluoresce. Okay, I'm showing in green color because I am using one fluorescent tag, that is FITC. It actually sends a green signal. This is the first step. Second step of this technique, so this is fluorescence. So we will actually fluorescently tag these cells. Now we will do another step that is called photobleaching. We will photobleach these cells. What will happen? Basically, a particular small region is actually bleached with laser beam. So we will throw a laser beam on a particular small area. Let's suppose here in this region we are actually throwing laser beam. So what will happen, from this region there will be no fluorescence coming because it will bleach the area. This region where there is no fluorescence tag is present or antibodies, we call this region bleached or irradiated portion of the cell. So what will happen, few lipid molecules will not actually contain the fluorescent tag here. Let's suppose these three lipids are not present, so there in this region there is no fluorescence is coming, so we photo bleach this area by using laser beam in a small area, laser beam. Now the third step of this technique is we will give some time to the cell to recover the fluorescence, recovery. So recovery time will be given. So that's why this technique is called fluorescence recovery after photobleaching. So after photobleaching we will see whether there is recovery occurs or not. So let's see graphically how it looks. Let's suppose this is the time scale, okay, and this is the intensity of the fluorescence of this area, bleached area, intensity of the fluorescence bleached area. At first, when we have not performed bleaching or photo bleaching, let's suppose the fluorescence intensity is here. It is showing the fluorescence intensity in this level. Then after photo bleaching, the fluorescence will actually come down. It will fall. Now two events can occur. One is there is no recovery of the fluorescence, or yes, recovery, there is a recovery. There will be two events can occur. If there is no recovery, the fluorescence will remain like this. No recovery means these lipids are not laterally diffused, can diffuse laterally. No recovery means there is no lateral diffusions or movement. So I am writing here no lateral diffusion. So they are immobile. No means that means this line will be shown by lipids if the lipids are immobile. Okay. But if the lipids are mobile, what will happen? The lipids that has the fluorescent tag antibody, they will diffuse into the bleached area, and the unbleached, these lipid molecules will diffuse away. So some will diffuse away and some will diffuse into this bleached area. So what will happen, after a few times you will find some molecules or lipid molecules are actually coming here. Now if the recovery occurs, then we will see the fluorescence is actually coming up and up. But what scientists have found that the recovery is not complete. So if the recovery is there, so I am writing here recovery. If the recovery of fluorescence is there, we will call that these lipid molecules are actually mobile. Now scientists have found that this graph, not this, so lipids are not immobile, lipids are mobile. So from this technique, fluorescence recovery after photobleaching, we can confirm that there is lateral diffusion occurs in the plasma membrane. Now I want to discuss some results of this fluorescence recovery after photobleaching. So I have to remove these writings. So what are the results scientists have found? I want to mention this. Okay. The first point they have noticed that all phospholipids, or phospholipids means all phosphoglycerides or sphingolipids, are freely mobile to a particular distance, like approximate 0.5 micrometer. They can move freely diffuse, laterally diffuse up to a distance of 0.5 micrometer. Most of the lipids cannot move further. So most of the lipids are restricted to longer distance. Okay. Now they also found that in the cell there is protein in the plasma membrane. There is protein rich domain is present. The protein rich domain, the protein rich domain, the diameter of the protein rich domain is 1 micrometer. Let me explain with the diagram of a cell. Let's suppose this is a cell. Scientists found that in the plasma membrane there are some portions or domains of protein rich regions. Let's suppose this region are the protein rich regions. Now they have found that in the plasma membrane there is also lipid rich regions are present. Lipid rich regions. Okay. Now the diameter of the protein rich regions is actually 1 micrometer, and the diameter of these lipid rich regions is 0.5 micrometer. And they have found that lipid molecules in the lipid rich region. So let's demarcate. This is the protein rich domain and this is the lipid rich domain. So they observed that lipids that are present in the lipid rich domain, they can diffuse into that particular area. They cannot diffuse from one lipid rich region to the other lipid rich region in a particular leaflets. Let's see. So they cannot move from one lipid rich region to the other. This is not possible because the presence of this protein rich domain, they are preventing the rate of, now this is very important, the rate of lateral diffusion in case of plasma membrane, that means natural plasma membrane. They found that the rate is 10 to the power minus 8 centimeters square per second. Okay, but in case of liposome, that means the artificial bilayer liposome, the rate of lateral diffusion is high, 10 to the power minus 7 centimeters square per second. As you can see in the plasma membrane, that also contains protein regions, the lateral diffusion is 10 times lower rather than the liposomes that are only made up of lipid molecules. So basically proteins actually prevents the rate of the lateral diffusions in case of plasma membrane. So in this video we have learned about rotation, flexion and lateral diffusion. In my next video I will talk about flip-flop movement in more details. Hope this video will help you. Thank you for watching and always keep learning.